All right, welcome back to DNA Replication Part 3. So, last time around, we introduced you to all of the players involved in DNA replication. Now we're going to put them together and build a mental map of how this takes place. So, again, remember we had originally DNA A binding to DNA A boxes that causes a bubble to form. Around that bubble, DNA helicase will assemble, and there will be one on each strand, and they will move in opposite directions. Now, what we are visualizing here is only one of those two replication forks. So the exact same thing will be happening in the other direction. So DNA helicase, again, moves along a single strand of DNA and it separates two strands. If you recall, this separating of strands will cause a positive supercoil to occur. So immediately in front of DNA helicase, topoisomerase 2, or DNA gyrase, you can call it either, moves ahead and uses the power of ATP to introduce negative supercoils. That way, when DNA helicase gets there, you've already relieved the pressure or the tension that would be introduced by the strands separating. Now, anytime you have naked DNA, single-stranded DNA, it will just simply adhere to what is complementary to it. So we have these single-stranded binding proteins that will come in and they recognize DNA in a non-sequence specific manner, and they prevent the two strands from coming back together. Next thing that happens is primase will come in and it will start laying down a complementary sequence of RNA. And this is a very short sequence. It's indicated here in red. So here in purple, we have the parental strands. The blue strands are going to be the daughter strands. And the red is an RNA primer. Again, the reason why we have this primase here is DNA polymerase, which is going to be adding the DNA, cannot just start from nothing. It needs a 3' end to add a nucleotide to. Since there is no 3' end present for DNA polymerase to add to, it could not start adding DNA. So consequently, we add in a little bit of RNA, again, only 10 to 12 nucleotides. And then onto the back of that, we can start adding sections of DNA. If you recall, we said on the leading strand, there would be a single RNA primer, and then we have the leading strand extending on. Notice here we have a second DNA polymerase. This can happen with just a single primer and one big long extension because it is moving in the 5', the 3' direction. Over here, we do not have that ability. If you notice, when we put down the DNA and start progressing, it is moved in order to move in a 5' to 3' direction. We are moving away from the replication fork. So we actually have to lay down primers in segments. We'll start off by laying down one, and DNA polymerase will start traveling along and laying a segment down. As the replication fork is moving this direction and opening up more, primase will come in and lay down another primer, and it will progressively just keep chasing this DNA helicase and adding in an RNA primer every once in a while. So on this side, we will start laying down a piece of DNA attached to that RNA primer. And when you hit a scenario where the RNA polymerase reaches a primer, what happens is the primer is degraded away until we are left with just DNA, but there will be a break in the backbone or a failure to connect the two pieces of the backbone together. At that point, DNA ligase will come in and repair that. So what we see here is actually there used to be a red primer present right here. It was degraded away by DNA polymerase. And then once the polymerase reaches the next chunk of DNA, it will stop degrading and fall off, and then DNA ligase will come in and join the ends together. So, wouldn't it be nice if this was the entire picture? Well, unfortunately it's not. There is in fact more. So there's not only one DNA polymerase. In E. coli, there happens to be 5 different polymerases. Now, each one of these polymerases has a specific type of function, and we will get into some of those here. The main ones that are in DNA replication are DNA polymerase 1 and DNA polymerase 3. Polymerase 2, 4, and 5 are all involved in the specific activity of DNA repair or replication of damaged DNA. Now, I will not specifically ask you to remember what the different types of polymerases do. So you don't need to memorize that polymerase 1 does what I'm about to tell you here in a moment. But what I do want you to remember and be able to explain is that there are different polymerases, and each one has a separate function. Some are involved in replication, some are involved in DNA repair. So, why do we need 2 polymerases to carry out normal replication? Let's look at their specific functions. So, DNA polymerase 3 is responsible for most of the DNA replication. It is a complex protein built of 10 different subunits. So 10 separate proteins produced by 10 separate genes come together and assemble to do this. The alpha subunit is responsible for catalyzing the bond formation between the adjacent nucleotides. And the others have other functions here. We have a table here where you can see all of the different parts and what they do. So here, for example, is the alpha subunit. It actually synthesizes the DNA. Here's another subunit, and you'll notice these are all in Greek letters. This is a very common convention in molecular biology. This is involved in proofreading. So if you accidentally put in the wrong nucleotide, this will fix it. This is an accessory protein for that function. We have the beta subunit, which is a clamp protein. It prevents the polymerase from falling off. So it slides back and forth, but it can't come off. Most of the other subunits are involved with loading the complex on and helping the clamp part work. So when you have all of the subunits together in a complex, we'll refer to that as the DNA polymerase 3 holoenzyme. So when you see the word holoenzyme, you can infer that it's built from many different subunits. Now, DNA polymerase 1 is just a single subunit, and that particular subunit will look a lot like the alpha subunit in DNA polymerase 3. DNA polymerase 1 has the specific function of when you encounter RNA primers, it will remove them and replace it with DNA. So essentially, DNA polymerase 3 will be traveling along until it hits the RNA primer. When it hits the RNA primer, it will fall off. DNA polymerase 1 will come in and replace the RNA primer with DNA nucleotides. All right, so let's take a look at the overall shape of the DNA polymerase molecule. And here we're specifically looking at a schematic of DNA polymerase 3, but they all have the same overall shape. What you have are a finger region, which wraps around one side of the double helix. You have a thumb region, which is wrapping around the other side. And this creates a hole in the back only big enough for perfectly base paired nucleotides in a perfect double helix to pass out of, right? So the DNA polymerase is moving towards the right side of the screen. Here is the template. Here's 5' to 3'. Here is the growing piece of DNA, 5' to 3'. So notice that we are adding nucleotides on at the 3' end, and that is making the strand longer and longer and longer and longer. How this overall process works is the nucleotides will come in, bind, it will be hydrolyzed, the backbone will be connected together, and it will continue progressing through the back. If you reach a point where the wrong nucleotide is in place, it won't form a perfect helix, and it will essentially create a little bulge. That bulge physically can't fit out the back. When that happens, the entire polymerase starts backing up. You'll get this growing 3' end fitting up into this pocket, and it will start chewing it backwards until it reaches the air, and then you will start moving forward again and put in the correct nucleotides. The overall shape of DNA polymerase helps explain the principle that it is unable to just initiate DNA synthesis by itself, that it has to be added on to the back of an RNA primer. Okay, so if we go back and look at this image, in order for this to work, it needs a 3' end for the new nucleotide to come into. It can't just take random nucleotides and just start base pairing them and linking them together. It needs a 3' end to grow from, right? So that is why we start off by producing an RNA primer. The polymerase that makes RNA has no problem taking just blank nucleotides and adding them together on the complementary strand. This is in fact very similar to what happens every time we transcribe a gene and produce a messenger RNA. We use a very similar type of activity. So we hijack that activity and produce a little RNA primer, and then we start adding the nucleotides on. Now if we start thinking about the problem of the 5' to 3', you can see why there is a lagging strand and a leading strand.